The next topic is what happens to the proton gradient if it gets uncoupled. So to remind us about the proton gradient, I have this diagram back. So the proton gradient is all the protons that are pumped out in this intermembrane space. They create that pressure or potential energy that is released when they flow back through, and that energy is used to make ATP. That's the chemiosmotic theory. So if that proton gradient is uncoupled by something called the uncoupling protein, which is a good name, uncoupling protein 1, it's also abbreviated UPC1. It's also called thermogenin. Both of those names are good. Uncoupling protein makes you think of uncoupling a gradient. Thermogenin makes you think of heat. Thermo means heat. This protein is naturally occurring, and it's found in the mitochondrial inner membrane, and it allows the protons to flow back through without going through the ATP synthase. So what happens in that case is that potential energy is still there, and instead of being used to make ATP, it's used to release heat. So that's why one of the names of the protein is thermogenin. Why is that important? Well, babies need a way to regulate their body temperature because they can't put clothes on themselves. So they have a lot of brown fat, which is high in thermogenin, and some of their protein gradient is uncoupled, and it's used to make body heat. The other place it's important is for animals. So animals like bears use it when they hibernate. And sometimes people have a genetic defect in their thermogenin and they have too much of it and they make very little ATP. Their bodies are very hot. Their body temperature is too high and they lose weight. So normally babies have this brown fat and thermogenin is there to help them with their body heat. But then as you grow and become an adult, a lot of that brown fat goes away and the thermogenin or uncoupling protein levels decrease. The other thing that can happen is you can have a poison in your body. Those are poisons that uncouple the gradient. One of them is 2,4-dinitrophenol, and that's a relative of the compound we used in the lab to do the enzyme assays. 2,4-dinitrophenol allows protons to go across the gradient, and if you have too much of this in your body, it uncouples the gradient totally. You don't make any ATP, and you're either very sick or you die. Okay, the next topic, last topic for chapter 11 is, this came from one of the homework questions and I thought it was a really good one, mutations in enzyme active sites. So there's a question in the homework for chapter 11 that asks you what happens in a certain enzyme if you mutate the one of the normal amino acid residues to a different one. So we'll just write that here. I don't have a picture of it, but the mutation was E189A. And what was important in this enzyme is there was a magnesium ion that was in the active site, and it coordinately bound to this amino acid. If you mutated it to an A, what happened? So I like this problem because it makes you think about metal ions that are important in active sites, but it also makes you think about amino acid structure and the one-letter code. So E is the one-letter code for glutamate. You don't have to memorize any of this because you can look it up. And the side chain for that was a carboxyl group. So it makes perfect sense that this would coordinately bond to a magnesium ion and help stabilize it in the active site. Well, what happens if you mutate that glutamate to an A? A stands for alanine. Alanine side chain is a methyl group. So do you think a methyl group would coordinately bond to a magnesium 2+ ion? The answer is no. So if you did this mutation, the enzyme would lose activity. So this would be a bad mutation. So the other way you could look at this question is, what would a mutation be that would not cause any loss of activity? And there's a really obvious one. Glutamate is almost exactly the same as one other amino acid. What would that be? Let's use a different pen. So if we made this mutation, E189 stands for the position in the protein. If we did E to D, we're going from glue to aspartate. There would most likely be no loss of activity. Those both have carboxyl groups and should coordinate the magnesium equally well. The last topic is mutations in enzyme active sites. I took this topic from a particular homework problem in the Smart Work 5 electronic homework for chapter 11. So in that problem, there was a mutation in an enzyme at position 189 from E to A. This particular enzyme had an active site magnesium ion that had to be there for activity. So the question asked, if you did this mutation, would the enzyme be less active or more active? So first of all, you have to figure out what amino acids are we dealing with. E stands for glutamate. You don't have to worry about memorizing that because you can just look that up. And the side chain for glutamate was a carboxyl group. It's one of the acidic ones. And it makes perfect sense that that would coordinately bond to a positively charged magnesium ion. So A, which would be the mutation or the mutated amino acid, stands for alanine. And the side chain for alanine is a methyl group. So do you think the methyl group would easily bond to a positively charged metal ion? And the answer is no. So this mutation would most likely have a big impact on the enzyme, and the enzyme would lose activity. Then for purposes of the test, think about a way you could make a mutation that would not have a big impact. What if you did this mutation? E to D. Again, E is glutamate. What's D? D is aspartate, and that has almost exactly the same side chain as the glutamate does. So most likely this would be able to coordinately bond to the magnesium 2+ ion, and this would have little to no impact. And this enzyme with this mutation should be almost as active as the normal enzyme. So this one would be an okay mutation. This would be a bad mutation.